Good morning, AP Bio. Here we go. We just wrapped up talking about Darwin and folks and theories of adaptation and change and descent by modification and how things evolve. Now we're going to bring it down more into genetic level, a micro level, than the macro, looking at fossils, etc., kind of thing. And so this chapter in your book is Evolution of Populations. All right, so let's start with some defining stuff. What's a population? It's a localized group of individuals belonging to the same species. Bam. What is a species? Group of populations whose individuals have the ability to interbreed and produce fertile offspring, viable offspring in nature. And we'll see as we go through this learning here that there certainly are different species who can reproduce, but they don't produce viable offspring, meaning fertile. Viable doesn't necessarily mean living, it just means fertile. Okay, the gene pool is the total amount of genes in a population at any given time. Okay. A locus is fixed if all the individuals in a population, homozygous. Well, okay, homozygous for the same allele, so it's fixed. And then the genetic structure of the population's frequency of alleles and genotypes. And we're going to talk here about Hardy-Weinberg equilibrium, and it's big, and one of the big statistical things in population genetics as they look at trends of changes in allele frequency and adaptations over time. Okay, now, one common stated thing, organisms don't evolve, okay? Organisms themselves don't evolve. Individuals don't evolve. Populations evolve. So, you know, the blue jay that I see out in my backyard right now is not evolving. It's just going along living, and right now it's in the, the bird bath. Okay. Doesn't mean it won't grow, doesn't mean it won't change, but it's not evolving. Evolving is change in a population over time. Okay. So species can evolve. Okay. Anyhow, and speciation we'll talk about later. Microevolution is that change in allele frequency in a population over generations. And we're going to look at it after we talk about Hardy-Weinberg. But there's three ways that allele frequency can change. Natural selection, which is the only one that can allow for adaptation and evolution. Okay, the only way you can evolve is natural selection. Genetic drift and gene flow can also cause changes in allele frequency, but they do not cause adaptation. In fact, and sometimes in many respects, they lower the allele frequency. They go in the opposite direction in natural selection. They limit populations and can be causes for extinction and stuff. Anyhow, moving on. So let's remember, at the heart of Darwin's theory is genetic variation. You have to have something to work on. You have to have something to select for. If there wasn't variation like diploid alleles with different versions of alleles, then you wouldn't have any variation and you wouldn't have natural selection and adaptation. So you have to have something to choose for. Okay? And that variation is caused by differences in the genes or other DNA segments. Terminology we should know a little bit from Punnett Square, but the phenotype is what you see. Well, if you can see it, but it includes, blood types and things you can't see. But if we just think about it, phenotypes are the visible types of hair color, eye color, influenced by both the genes and the environment. Okay. Natural selection can only act on that variation with a genetic component. All right. So adaptation evolution does not create new alleles. It can only work on the ones that are there. And if you don't have multiple alleles or multiple choices, then it has nothing to select for. Okay. And we saw this way back with genetics. but you have both discrete and quantitative characteristics contributing to that variation. Discrete ones are the either or purple-white color we did a lot of Punnett square with. Quantitative, and we talked a little bit about average heterozygosity back then a little bit. That's like those shades, those skin tones, those fur color things, those norm of reaction kind of bell curve things that even eye color to an extent because the number of genes on it, that range of stuff. So it's not an either or trace. Okay. Genetic variation can be measured as gene variability or nucleotide variability, and average heterozygosity is just the average number of folks that are heterozygous in that population. Okay. And so how do you check that variation? You compare DNA sequences. Okay. Now, how do you notice? There's different ways to have genetic variation. You can have it just within that population and species. Okay, so polymorphism is just having those discrete forms, whether it's two or more things. This is the purple-white kind of coloring thing. This is AB, ABO blood types. This is, do you have freckles or don't you have freckles? Okay, that's a within. That's not a, that's within that population, within that species kind of thing. There's also variation between being male and female, sexual dimorphism. So not just the genetic, but that decision of male or developing as male or female. And also in that, and this will be something we talk about later, the differences between, it's the type of reproduction that you might have. Is it for that species intrasexual where you have, this is the rams or deer and things that you see locking horns and button heads and deciding who's the alpha and who gets to mate with the females. That's what intrasexual is. It's that competing as to who's the, for lack of a better term, strongest male, which indicates the strongest genes and the one that gets to mate with all the females. And while that's the biggest example generally because the males in many organisms are the larger type and more physical and whatever, but there certainly are plenty of other organismal things where, it would be the quote unquote females also. So that's the idea. Intersexual is also called mate choice. And that is when you see organisms and animals where they have a mating ritual, a dance, a call, a song, or something like that, and they need to attract. It is one of the reasons why you see this in bird coloring, like right now as the cardinals come back. The males often in many like bird species are very colorful. Big red cardinals, peacock, the feathers in peacock tails and stuff like that generally favor one species, and oftentimes it's the male because in a sense the female is carrying the egg and giving birth and everything else, so it's the female's choice. Okay. In selecting, so you have those big colorful males or the songs or the dance or the whatever to attract the female and say, hey, I've got the best genes and you want to reproduce with me. Okay. And then there is variation. We'll get into more examples of this, but the biggest one between populations and what can create variation, geography, because, well, it's geography. So here, and I stole this from your book, you're looking at the Sierra Nevadas, which if you've never driven through them, they're beautiful. But on one side, as you come and say you're coming from, as I have done many times going when I was driving my son back and forth to Northern California, you go through Utah, Nevada and stuff, and it's all desert, and you hit the Sierra Nevadas and you hit the mountains, and on the other side is all lush and green and everything else. But you can have different species of the same kind of plant, and they differ because they're isolated. So they have, and this is why we're going to talk to more of these things later, but that's part of the whole speciation is as they get isolated by geography, then one, you can see a species diverge into multiple species, and two, they will have genetic variation to be able to withstand the environment they're in. So on this side, the hot, dry, deserty climate, and here the more wet, on the Sierra Nevada range, the more wet, greener climate. Okay. But here's how where we want to go today. We want to talk about Hardy-Weinberg. Do all populations evolve? Kind of yes. Hardy-Weinberg is a theory. I equate it to, if you think of chemistry, the ideal gas law. There is really no ideal gas, but many gases behave like an ideal gas, except at high pressures and low temperatures. Hardy-Weinberg, in theory, is making the assumption that a population is fixed, it's non-evolving. And so it has some stipulations say this population is not evolving. And so you can do that. And are there examples? There certainly are examples, and we'll look at one here. But again, it's a basic theory to show that genetic variation and changes are occurring and are easy to track. But you need basically a null hypothesis to start with. So Hardy-Weinberg describes the genetic structure of a non-evolving population where the frequencies of alleles and genotypes are stable over generation, never change. In order to be non-evolving, you got to meet 5 conditions. Number one, it's got to be a very large population size, and where you see things now that appear to be actually hitting Hardy-Weinberg equilibrium, it's because we're a much more global society. It has led to allowing some of that, because that's really kind of, in many respects, the type of population size you need, especially if you're looking at human stuff. You need to be isolated from other populations. Isolation from other populations, no gene flow. Okay, so you got to have a large population size, and you can't have entry of new genes, and we'll talk about gene flow in the future. There is no net mutations, meaning yes, as things mutate, but really overall in the long thing, there's mutations, but whether they don't reproduce, whether they don't survive, whether they are just a chance and the organism doesn't make it, something along that line. But overall, while you do have mutations, in the big scheme, it's no net mutation because it doesn't shift the statistics. You got to have completely random mating. Okay, so that kind of puts away what we were just talking about. That sets aside the intersexual choosiness or alpha male fighting stuff, that is absolutely completely random. And that there is no natural selection. That there is nothing to choose. There is no benefit. There's no choosiness. There's nothing there. There's no function on natural selection. Because again, you're not getting any change. You're not mutating. You're not getting any new genes brought in from an outside population. So and again, the heart of Darwin's theory is you got to have something to choose for. If you don't, then you're not going to have adaptation. Okay. So as you can see from those, they are, this is the reason it's theoretical. Now, the population size, that is getting more, again, as things are becoming more global, and it's not just us, but as people have become more global, more plant species and animal species and things have been brought and introduced to areas and stuff like that. So while there's still gene flow and stuff, as this happened, we got the population size, started kind of knocking back some of that gene flow, but no net mutations is hard. Random mating is almost impossible because there is some, it's basically impossible. There is selection there, and natural selection is working on stuff. Anyhow, here's our formula. So it's just a calculation. P squared plus 2pq is q squared equals 1. So P is generally designated as the frequency of the dominant allele. So P squared is just the frequency of the diploid. Q is the frequency then of the recessive allele. And so Q squared is the diploid. And then 2PQ is the heterozygotes. Okay. For one single population, smallest unit of evolution. So the frequency of the dominant allele plus the frequency of the recessive allele will always add in heterozygotes, but it will always add up to 1. Okay. So the frequency of an allele in a population can be calculated for diploid organisms. The total number of alleles at a locus is the total number of individuals times 2. Total number of dominant alleles is 2 alleles for each homozygous, 1 for each heterozygous. You use the same logic for recessive alleles, and you'll see that. By convention, if there are 2 alleles at a locus, P and Q are used to represent the frequency. So P plus Q is always 1, because you can't get over 1. All right? You can't have more than what's there. And that's the math that helps us get going. So here we go. Here we have red and white population. We have a total of 500 plants. 320 of those are homozygous dominant. So that means you have 640 alleles. Okay. 160 of those were heterozygous. So you add 160 dominant R alleles. And so out of the total 1,000, because you had 500 plants and each one was diploid, 800 of the 1,000 are the dominant big R allele. So that's 80%, or 0.8. Using the same logic for the recessive, then you had 20 homozygous recessive, which is 40 of the recessive allele, plus the 160 from the heterozygote is 200 over 1,000, so that's 20%. So 0.8 plus 0.2 is our how we get to 1. That's our allele frequency. Okay. And then for Weinberg, the P squared, 2PQ, Q squared. So if P is 0.8 and Q is 0.2, then you can do the math. P squared is 0.64, 64% are homozygous dominant, 32% are heterozygous, and 64 or 4% of the alleles are recessive. Okay. Homozygous recessive. All right. So Hardy-Weinberg principle states that the frequencies of alleles and genotypes in a population remain constant over generations. In a given population where gametes contribute to the next generation randomly, allele frequencies won't change. Okay. And Mendel inheritance preserves that variation in a population. And we can go back to, why do you have heterozygous alleles like sickle cell still, or the heterozygosity of having sickle cell still in the population, and that goes to Mendelian inheritance. So again, it's a theory of a hypothetical population that is not evolving. In real populations, allele and genotype frequencies do change over time. So like I said, those things that we said, those 5 requirements of what are happening, kind of hard to do. So that's why Hardy-Weinberg is a theory, not a fact, because populations do change, but you can predict population changes, stability, etc., by using Hardy-Weinberg. Okay. So let's just apply to one sample, and that'll work for today, and then we'll move on to more tomorrow. Okay, PKU is a very low gene mutation rate. Okay. And it is one of those examples that now that we've gone worldwide, PKU, which is a phenylketonuria, it's just a disorder that if you follow dietary restrictions, you're fine. Anyhow, it is has a very large population. The population is basically the world, because it is not like some allele frequencies or recessive alleles. It is not relegated to certain populations, ethnicities, creed, whatever. Okay, it's worldwide, so there's no migration, there's no gene flow anymore. because it has, and it has been worldwide for deal. It's one of those, again, that just, it's one of those mutations that didn't really change much, because if you watch, if you follow the diet, you're fine. And so you can reproduce and it can go, and it doesn't select for any one population over the other. So there's no net gene flow. Most populations have same frequency. The population is basically the population of the world. Its mutation rate is slow, so that's close to the known net mutation. The mate selection is random because it's just whether you have the gene or not. And again, since it's not favoring one ethnic group or social group at all, it is completely random. Okay. Natural selection can only act on rare homozygous individuals who do not follow dietary restrictions. So again, that's the other. Now, serious health concerns for them and a shorter life, but it's so easily fixed with diet that it really doesn't change much. All right. So the occurrence of PKU is 1 per 10,000 births. Okay, it's again a rare homozygous recessive. So Q squared is 1 in 1/10,000th, or Q is 0.01. So then the frequency, if P plus Q equals 1, then 1 minus Q is 0.99. Okay, for the 99% homozygous dominant. And the carriers, the heterozygous carriers, 2PQ, 2 times 0.99 times 0.01, is about 1.9 or 2% of the US population, and that doesn't change much worldwide. Okay, so when you're looking at the fact that really you have 2% carriers, you got, 99% of folks, actually 99, percent of folks, even the heterozygous, are fine. Everybody's fine, actually. But at 99% homozygous dominant, and that's the same it's been for generations, because there's really no mutation on it, and it really isn't harmful unless you're not eating the diet. It has really landed at Hardy-Weinberg equilibrium for the most part. Not perfect, but it's basically there. Anyhow, that's how you use it to look at allele frequencies and changes and adaptations over time. We'll stop there with this. We'll move on tomorrow to talk about natural selection, drift, and flow. But for now, we're good. Stay healthy, have a good day, and we'll talk to you soon.